a good act to follow. So, hi, my name is Stephanie Adams. I'm the plant health care leader at the Morton Arboretum. A little bit about my background. I've been a certified arborist since 2007. I've been a board certified master arborist since 2017. I've worked in the tree care industry for about 23 years, and I also have a PhD in forest tree pathology. So I'm a plant pathologist, so anytime I say, hey, I got something really cool to show you, most people will be like, that's really bad, how can you get excited about that? And it's just kind of pathology humor. So yeah, the one thing that I really want to focus on that I think has been under discussed and underrepresented, especially when it relates to tree health, is the status of fine roots. A lot of times when we talk about roots and root health with trees, we're talking about the woody structural roots. We're talking about where they shouldn't be circling, they should be at or slightly above grade. We know how the woody structural roots are supposed to look, but we haven't really been talking about what the fine roots look like, right? And so this entire presentation is going to be going over that. So to keep in mind, kind of a refresher on tree roots, is that tree roots grow, most of them grow in the upper 16 inches of soil, especially here in northern Illinois. There are some species that do have, at maturity, do have tap roots. A lot of times those are in sandy soils. I know some exist, like eucalyptus in Australia. They have tap roots because they can get down to their really deep water table. But in northern Illinois, upper 16 inches of soil is just the general rule. And even that, usually it's below 3 inches, so they usually don't stay on the surface because they need that moisture and they're not going to compete with turf grass and weeds for soil space. And so they grow at that 16 inches because they need water and oxygen, which Michelle already touched about. And so the thing is, in clay soils, because they do need water and oxygen, they're even shallower because they're basically growing in a brick, right? And so because of that feature, they are more susceptible to drought. So when it dries out, we know clay soils really dry out, and they're really difficult to re-wet. And then it also makes them more susceptible to drowning or flooding, any excess water. And so like what Tricia mentioned, and I'm going to talk about this a little bit more in my next presentation with oak decline, is that we are experiencing a period of climate change. And I'm not going to go into the philosophical debate of how it happened, but we are here, period. And we are experiencing more precipitation, and that is affecting this system where you have shallow roots needing reduced vigor and lifespan. Healthy fine root system is largely overlooked, and that's what I went out actually just before this. I don't have the nice setup that these two have because I just want to show how simple it is to look for fine roots. You don't need equipment, you don't need anything super fancy. And we need to start identifying what do the roots of different tree species look like. But for the most part, whether they're tree roots, they're tomato roots, they're houseplant roots, for the most part, they're going to be kind of a creamy white color. It can be vary within species, like river birch, they will have exfoliating, the peely orange bark. If you dig up a persimmon, they actually have naturally black roots. Which for the most part, when I think of black roots, I think of unhealthy roots, which I'll get to in a minute. If you dig up mulberry, which I have over here, they have yellow roots. So yeah, there's always kind of competition of whether you can identify trees, especially winter ID is always a little bit tougher. I'm even bad at identifying buds. But I don't know, if you can't identify a tree by its roots, like do you really know how to identify trees? But so the other thing is, so the roots, if you roll them around on your fingers, and please, go ahead and manhandle these. I just went over here to the park and dug them up. You should be able to, they're firm, they are not squishy, they don't collapse. There's a fine distribution or an even distribution across that root ball, and roots for the most part are round. So if you have roots that are black, brittle, squishy, or they look like shoestrings are real flat, you have an underperforming root system. So this is a magnolia root system. Has anybody ever smelled magnolia roots? Oh my gosh, it's so wonderful. Like, seriously, treat yourself. Go out and dig up a handful of magnolia roots. And you don't even have to smash them up, just smell them. I'm not trying to trick you either. It's not like a persimmon, I swear. But Smell magnolia roots. You will enjoy it. But notice here, like this is a really healthy root system. It's evenly, it's white, it's that creamy white. But two, I lost my train of thought. Sorry, I lost my train of thought. So we have to understand what is proper fine root biology, because we need to know how they function. Why do we care about fine roots, right? We've been taught about how important the structural roots are, those woody roots. The thing is, whenever we talk about secondary problems and stressed trees and two-line chestnut borer, the thing is, these fine roots, those are the roots that actually absorb water and nutrients. If you can't absorb water and nutrients, your tree is going to malfunction. It's like, oh, you know, the human has a mouth, we just sewed it shut. I don't know why it's struggling. It has a mouth. You know, if it's not getting water and nutrients, it's not going to perform well. So they do arise from perennial roots. So perennial roots is one way to say woody structural roots. So perennial, just like with plants, you know, they will maintain those roots over time. They're not a short-term roots. Most fine roots are relatively short-lived compared to woody roots, right? They'll live for a couple months to a couple of years, but for the life of the tree, they will actually turn over faster. They're responsible for water and nutrient absorption. This is where your mycorrhizal associations are. So if you're adding mycorrhizae to a tree, hoping that it's going to help its life, but it doesn't have those fine roots, I mean, you're adding organic matter, which is good, but you're not actually benefiting that tree because it doesn't have the roots to make that association. So fine roots too, they are high in nitrogen. And the reason why that is because there's a lot of activity going on in those roots. They are the primary absorbers of nitrogen. And the thing is, in nature, nitrogen is limiting for everything. You know, we put down a lot of nitrogen, like people will fertilize blindly, which don't blindly fertilize. Do foliar and soil nutrient analysis and see what that tree actually needs. Because adding nitrogen will a lot of times make health problems worse. Sure, you'll see that the tree will green up, it'll put on more growth than normal. But the thing is, nitrogen is also limiting for fungi, bacteria, insects, mites, all those. So not only are you feeding the tree, you're also forcing it to put on new succulent growth, which is more susceptible to infection and infestation. And you're also feeding the fungi and the insects. So you can actually make, like if you want to fry out a blue spruce or a spruce that has rhizosphere needle cast, give it a nitrogen. You'll fry that tree out like that. So on mature trees, and the roots that I'm going to be talking about are the roots that are rarely bigger than a ½ millimeter. So these are the really, really small roots. And so this is a really great paper I found. I really fall in love with this image. And so it illustrates the different orders of fine roots. So I'm going to skip ahead for a second. So there are 5 orders of fine roots that are at least illustrated in this paper. And so the finest roots that I'm mentioning are the first and second order of roots. So they have a high absorption capacity, they have high respiration, and high nitrogen. Okay? And you can see, it doesn't have, so this is the epidermis of the bark of the roots. You can see on here, it's really relatively small. And that's because osmosis is happening. Water's crossing over that barrier in order to get into the roots. And so it doesn't have much of a physical barrier from the outside world. It has high nitrogen, so it's really favorable to any kind of microbes that can get into it. And there are a lot of soil-borne pathogens, which I'll get to in a minute. And because they are relatively short-lived for the trees don't really invest a lot of immune system resources to it. So trees and plants have 2 immune system responses. One is a primary, the other one is secondary. One primary example is resin production in conifers. So, you know, if you cut into a conifer, you're going to get resin oozing out, right? That is a conifer's primary immune system response. It's relatively evolutionarily low on the totem pole. It's not very sophisticated. It's, hey, if you try to invade me, I'm going to like push you out and like coat you in resin. But all trees have primary immune system responses. And so, and then the secondary responses are a lot of times the essential oils that we use in our lives to, you know, get different benefits. Their primary function in nature is they are plant-developed pesticides. Those are secondary immune system responses. And in order to produce those, you have to have water and nutrients. So if you don't have water and nutrients, it's not going to have the secondary immune system, it's just going to make it more susceptible to other problems. So you get into the third and fourth and fifth order roots, and these are the roots that actually take the water nutrients that are absorbed here up into the tree. So they have high transport capacity. So what this looks like, highlighted in red in all of these are the finest roots on all these species. And so when you dig up a root, and this is where we need to create this baseline of like, do we know what healthy roots look like? You know, if I said, imagine you had a tulip tree root system, which if those are the finest roots they have, those are huge, right? When you compare it to, I mean, even a maple or the walnut, those are really dense, big roots. And so if you have a hickory that has a root system that looks like a tulip tree, you have a serious problem. That would be an underperforming root system for a hickory, right? And so if you don't know what's normal, you can't identify what's abnormal, right? So that's one reason why I collected these samples. So I have over here a Scots pine, mulberry, cottonwood, green ash. And the ash too is really nice that it actually, you can see kind of side by side where these roots, where those fine roots aren't. And if you don't have those fine roots, you have to start asking yourself why. And keep looking a little bit closer. This, I can't remember what species it was, but this, you know, there's no fine roots on these. They're actually a little collapsed and kind of soft and squishy and brittle. So this is not a healthy root system. The maples, I always, you know, imagine a tulip tree and then imagine maple. Again, if you dug up maple roots that had tulip tree root system, you have a sick tree. That's a problem. That needs to be addressed. So, then the third order of roots is highlighted in green. So those are the roots that move it up to the fourth and fifth order roots, and that's what takes it up to the tree. So when you go out and you assess a site, and if you have a tree that's starting to have some of that staghorning, no matter what species it is, this is what Tricia mentioned, if you start seeing that staghorning, especially if it's pretty uniform across the canopy, you need to look at the roots. Because if you have staghorning that's uniform across the canopy, you have a problem downstream somewhere. And all this tells you is that for some reason water and nutrients isn't getting to that part of the tree, right? So if it was just one branch, if this was just showing symptoms, I would say, okay, look for a canker a little bit further down on the stem, right? Because it's girdled for some reason or something. For some reason, water is not getting to that. So people, they'll send me photos and be like, hey, what's wrong with this tree? I'm like, for some reason, water and nutrients isn't getting into the canopy. You're like, well, what could that be? I'm like, oh, that's like a shopping list, which I'll get to. So, and especially if you have uniform symptoms across the canopy, either it's an advanced vascular wilt disease, you have a root rot, because if you have a root rot, that's going to affect the entire canopy, right? So if you have the entire canopy, you need to look to an area that's supplying the entire canopy. So this is just shovels. So this is a white oak root system. So the one thing that's really neat with this is that these little yellow flecks that you can kind of see, those are actually the mycorrhizae, which is pretty cool to see that. So you can see mycorrhizae and kind of assess the health this way. I would actually consider this to be an underperforming root system for an established oak tree. It has some of the fine roots, but they're not really extended off the fourth and fifth order of roots, and they're kind of lacking in some areas. But they have the mycorrhizae association, so that's pretty good. So in order to really understand the whole importance of why fine roots are so important, you need to understand why trees need water. Trees do not need water for growth, for photosynthesis, all that sort. The majority of the reason why trees absorb water and there's transpiration that draws it from the roots up the stem and out the stomates and the leaves is purely to keep those stomates open so it can get CO2 so it can photosynthesize. That's the number one reason. Yeah, 96% of all water that moves through that tree absorbs, passes straight through it. And it's just to keep the stomates open. So if you have periods of drought, and that tree is actually kind of making the metaphorical debate of I can close my stomates to prevent water stress, but then I'm not going to photosynthesize. Or do I leave my stomates open and get drought stress, but it can photosynthesize? So that's why watering is the number one thing you can do for your trees, or actually having appropriate water. So you don't water blindly. And so that's why it's so important. That's why it's so important in order to have these fine roots, because if you want that tree to grow, it needs food, and it makes its own food, and it needs water to do that. Only 3% of the water that goes into trees is used for growth and 1% is used for photosynthesis. So when you have a root rot, and, or if you see a symptom, so even when you go to garden centers or go to nurseries or anywhere that sells plants, or even in planting beds, like if you plant somebody planted a whole bed of like petunias or something, and there's a lack of uniformity across that crop, then you have a disease, you have a problem somehow. It could be an insect, it could be a disease, but nonetheless, there's an issue going on. Because if they're clones, if they're cultivars, they should be uniform, right? They're produced the same, they're all cuttings, they're genetically exactly the same. They should look exactly the same. So, one, don't buy those. There's a lot of truths, and I use a lot of parallels between human healthcare, veterinary healthcare, and plant healthcare, because it's kind of the same issue all across the board of, actually having diseases and having problems. That's normal. We know every human in this room has gotten sick at one point in your life, right? You've had a cold. Being sick is normal. Not dealing with it, not managing it, that's the problem. And denying that you have it. If you go to somebody like, oh, I don't have any disease, don't buy their stuff because they're not looking and they're just like glassing over that they have problems. So the thing is, at the nursery, it's an assembly line. They process so many plants. And they do the best they can. They have a lot of sanitation protocols. They really do their best to produce good plants because they want to see good plants. They want their customers to be happy. But it's also just a difficulty of working in an industry where you're dealing with thousands of plants a day. And so if they potted up this plant when it still looked pretty good, or even this one when it still looked pretty good, and they had it on their potting bench and they processed 100 more, you can assume all 100 of those that followed that is now infected. So if you wanted to buy these, and you're like, I'm just gonna pick that one, and that one, and that one, look, because it looks pretty good, I would quarantine that or have it diagnosed or treat it with fungicides before you put it in the ground, because you don't want to inoculate your native soil. It's a lot easier and cheaper to treat a plant in a container than it is into our native soils. But prevention is true all across the board, whether it's human, veterinary health, or plant health. If you prevent it, an ounce of prevention is worth a pound of the cure, and fungicides are pretty expensive. So in landscape plants, usually the first symptom that you see that a tree is stressed or having problems, in this case root rot problems, is you start seeing that staghorning. And again, it's uniform across the canopy, so this is a white oak. And when you have that, when you have a client that has that, and I'm going to mostly speak from the landscape side of things, not the forest health side of things, because that's managed very differently. So when you see this, and it's not a, hey, let's wait to see what happens next year. This is a one-way. How many people have seen trees like that recover? Yeah, how many have seen them continue to decline? Yeah, so this is a one-way street. When you see this little bit, when those little 1/2 inch and 1 inch branches are starting to staghorn up the top of that tree, that is a big red flag going, oh my God, I'm going to die in the next 7 years. So you need to take another close look at that. So this, you see the staghorning across more than 80% of the canopy. You start losing some of those fine branches because the leaves aren't going to be growing there. We've learned from EAB that trees will take nutrients up as high as it can, right? You get the water sprouts that are coming out the stem with EAB, because above that point it's all girdle, so it can only go so high. Trees have a wonderful and really sophisticated survival instinct, like they want to grow. And so they'll start killing off, or those branches will start dying off because they can't get water and nutrients up there. And then, so those twigs will start dying back. In the later stages, you have none of those fine twigs. That canopy is really, really thin. Most of the growth is on those main stems, and it'll continue to decline. So the stage I refer here, I'll refer to again in the oak decline presentation. So most leaves are growing off the main or lateral structural branches. You start getting that staghornings, really bad. Especially you start seeing those 2 to 3 inch branches popping out that top. And then a lot of times you'll start seeing suckers up the stem, and the leaves will start discoloring. And at this point is when you start seeing the hypoxylon canker and two-line chestnut borer. I would say 80% of all cankers on plants and trees are secondary. They are stress related. One exception is like chestnut blight. That's a primary pathogen. But if you have hypoxylin canker, the hypoxylin canker will definitely take that tree out. It'll be the final straw. But that's not what killed it. It's not what led to this point. It is taking advantage of a tree that doesn't have an immune system, so it can't defend itself. Same with two-line chestnut borer. If you're treating trees for two-line chestnut borer and not actually working to identify the primary underlying problem, you're kind of wasting your client's money. It's a little bit malpractice, because if you take care of that primary problem, two-line chestnut borer will go away by itself. You know, sure, treat it for a while until you can find that stressor and you can mitigate that, but then you can phase it out. So when you're seeing those symptoms across the canopy, you know, again, it tells you that for some reason water and nutrients aren't getting to the top of the tree. And so we, as an industry, have this problem where we think we can diagnose things just by looking at it. And so again, as a background in plant pathology, and I've worked at the Georgia State Diagnostic Clinic and the Florida Diagnostic Clinic, I can guarantee you, you would not want to make that bet if you try diagnosing things just by looking at it. There's many things that share symptoms. So we need to get away from treating symptoms and start treating a diagnosis. And the nice thing is that you know you're going to be using pesticides prescriptively, so that it's more environmentally sound, and you're going to get better return, and your customers are going to be more happy because they actually see results. And so, you know, for example, I went out with an arborist who was treating peaches for peach tree borer. He's like, oh yeah, we've been treating for 5 years and they're still declining. If it doesn't recover and start getting better within a year, it's not the right treatment. Period. Don't keep doing it. And so I walked up to it and I'm just like, oh, here, there was a mummy, there's an old peach on the tree. I'm like, no, this had brown rotted stone fruit. This is Monolidia verticulata, it's a fungus. Like, it didn't have peach tree borer at all. And so for 5 years, they wasted their clients' money, and they're still going to lose the trees. So problems that look, you know, that cause that canopy decline are all these. And I just looked at the time, and I know I only have a half hour, so I'm going to go a little fast, but slow down on the important part. So just keep that in mind. There's many reasons why water cannot get to the canopy. So during stress periods, all that secondary metabolism, that immune system responses completely go offline. They're not even functioning because they don't have the water and nutrients to make those essential oils to protect itself. And so, so the stomate's closed, reducing photosynthesis, and then you get to the point where you actually get air seeding into the tree. So I suspect, this is just a hypothesis, that when you start seeing that staghorning, it's cavitation, it's air seeding, so it's actually in the vascular system, in the xylem, which are kind of like straws in many ways. And so you actually get air pockets in that xylem because there's not water in order to move within the system. And so the interesting thing about the air seeding and the cavitations is that, one, 93% of all trees in the world will never recover from that. That is now dead xylem. It is no longer functional. And so that part of the tree is now dead and gone. So you need to have, the tree has to be vigorous enough in order to recover from that. But the interesting thing, especially when you think about just general ecology, is that insects use a lot of different senses that we can only imagine in order to find host plants. And so a lot of communication in nature is actually chemical, so it gives off different gases to communicate. Well, the air seeding, and you can go on YouTube and look up tree air cavitation acoustics, and you can actually listen to it. It sounds like, and that's what air seeding, and the beetles can hear that and they're attracted to those trees, and so then you get the two-line chestnut borer. So disease roots. Usually they're black and brown in color, they lack the fine roots. The bark will be sloughing off, or kind of degloving, so when I say degloving, like literally think you're just sloughing off the outer surface of the roots. A lot of times they're flat and brittle and they lack volume. So this is a 3-year-old, this is a Georgia oak, which I know is a weird species, but this is a 3-year-old tree, an oak tree. It's native to Georgia and Alabama, but compared to this, these were collected at the same time, they're potted up at the same time. This has a pretty good looking root system. This roots are sad. So if you, again, anytime you buy a plant, pull it out of the container. You're buying the entire plant. Like, would you buy a house without looking in the basement? No, you're buying the entire thing. Whether it's walled and burlap or container, you're buying the entire plant. And most people in this room, we're planting trees for the longevity of that genetic potential of that tree. Like if I plant an oak, I want it to live for 300 years, right? I'm not a homeowner and some landscapers or whatever. I'm not, we as arborists, we usually come in at that 15, 20-year mark when they're starting to decline because they weren't planted correctly. And it's like, I don't think in that time frame. I think about the longevity of that species. So always look at the root system. So the neat thing is, and this is in containers, is that if you identify these disease problems and actually treat them with the fungicides that they need, which I'll get to in a minute, the roots will start recovering. So you can see this is the new growth. The fungicides, the treatments, like they actually protect the newly emerging roots. They'll never cure and save the roots that are already infected, because once these fungi are within plant tissue, they're protected. You can't get to it. But it will help the root system recover if the tree has enough energy to do so. So healthy roots are nice and firm, they're round, they're creamy to they're white to creamy white, and they smell nice and earthy. Has anybody ever dug up roots or even repotted your house plants and you unpotted it and it smelled terrible? Like it had that sour smell? So that is the smell of bacterial soft rot. That tells you that there's too much water and poor drainage. And so the nice thing with bacterial soft rots is usually if you can improve drainage or reduce watering or get into a better drained soil, it'll recover on its own. You don't have to do anything for that. But that also tells you that you have no oxygen in that soil. It's gone anaerobic, which Michelle mentioned. So this kind of side by side, so this is a diseased root, so it's brown, kind of collapsed. You can see the bark, you can actually kind of see it's kind of puckering, like shriveled a little bit, and you have a nice creamy white root system. So there are dangers of diagnosing things just by looking at it. Number one, you might be using the wrong pesticides. So again, we as an industry, we like to lump things. We are definitely lumpers, not splitters, when it comes to information, because it is. There's so much information, especially when you're working with living organisms. We have a lot to work on. And so we tend to lump things like all root rots are Phytophthora, right? I know that's a prevalent mindset. Just like all declining oaks must have oak wilt, right? All declining elms have Dutch elm disease. But there are two other diseases that look like Dutch elm disease and American elms. And so you can't identify the difference between those in the field. You actually have to culture it out. The other two are verticillium and phytoplasmic diseases. So if you might be using the wrong pesticides, there's the potential of spreading it. So if you're dealing in a landscape and you know you have a diseased plant, if you don't have that identified, it's nice to know you go into your client's property, you're like, okay, this tree was diagnosed with Rhizoctonia root rot 2 years ago, we treated it, but we know that these are all the other host plants in your yard. Make sure you sanitize your tools before digging around those. Just like sanitizing your pruning tools, sanitize your shovels, sanitize your boots in between properties. You need the other nice thing about diagnosing it, having properly diagnosed, is that you can identify if it's a primary or secondary problem. If it's a primary problem, yeah, you need to spearhead that and get on top of it. If it's a secondary problem, you need to keep investigating to figure out what the primary problem is. Because secondary problems are usually weak pathogens or weak insects, and they can't attack healthy trees. So if you take care of that primary problem, the secondary problem will resolve itself. It also helps you understand sanitation measures that you need to take, especially in between properties. Like we all know, or we've seen people who, especially, I'm going to pick on people who mow lawns, which we need those people. It's a great industry, we need it. It's a lot of jobs to a lot of people, but it's like, they don't sanitize their mowers in between clients, which they can't. It'd be impossible. But we all know that, you'll have a homeowner and be like, oh, I always had like this uniform species of grass in my yard. And things keep creeping in their yard, and none of their neighbors have it, and it's well distributed because it's coming in on the lawn mowing, or the lawnmowers. And so it also helps you identify where the pathogen might have come from if you did introduce something. So this is a little bit of a show and tell. This is me being a little salty, I'm not gonna lie. Because we have this tendency of diagnosing things just by looking at it. So this is your quiz of how accurate are you able to diagnose things just by looking at it, right? So what do you think this disease is? These are arborvitae that are right outside our children's garden, right at the entrance. So the symptoms you're looking at, these two trees have been replaced. This one's starting to show symptoms, starting that yellowing. And even this one is one thing. know your own body and what colors you can see. Just like some people don't have sense of smell. I know somebody that smoked 3 packs a day has no sense of smell anymore. It happens. Not everybody can see the same colors. And so for me, I can see that this tree is not as green as this one, right? And it has that little bit of yellowing. And if you're going to manage this disease, you're not going to manage at this point because that's too far gone. You're going to manage at this point and try to get that plant to recover, right? So if you know that you have a color spectrum that maybe you can't see very well, take photos and ask people like, hey, which one's greener? And they'll point it out to you. I've had plenty of bosses that did that. It's kind of amusing. But so they replaced them. They had these trees, they started declining, they replaced them. And the one that they replanted is also showing symptoms. And whether that came from the nursery or it's because there's something in soil, I can't tell you at this point. But the roots had the bark sloughed off, so you can see this is actually the steel, this is the inner part of the root. And the bark, this is actually the bark that kind of slid down the roots. So this is Rhizoctonia root rot. And Rhizoctonia, especially on arborvitae and yews, I've seen a lot. And I think it's because a lot of nurseries will assume it's Phytophthora, they'll treat for Phytophthora, and so all these true fungi are getting by going, ha ha ha, I no longer have competition. So this is a little ginkgo, kind of a unique cultivar that we got in. I took it out of the container, this has been in the container for like a year, and when I pulled it out of the container, the root ball completely fell apart. And so if you have a healthy root system, that ball should be pretty solid, right? So that automatically tells you, it has an underperforming root system. You know, maybe if it was potted up a week ago, okay. But the roots are pretty soft and squishy. Magnolias, or ginkgos do have kind of a coarse root system. And so this was actually Phytopithium, which I'd never heard of this before. This is pretty a new species. So the genus of Pythium is starting to be divided out into new genera. But yeah, this is Phytopithium root rot. So we got a bunch of arborvitaes in. So for the most part, the canopy looked pretty good. But the difference is you need to know the difference between healthy looking and disease-free. Just like people in this room, I'm not going to pick on anybody in particular, but it's like you can say, look at me, I look healthy looking, you don't know if I'm disease-free, right? Same applies. And yeah. There's another joke in there, but I don't know. I don't have time for that. So it's healthy looking, but this is why you need to take it out of the container. This is healthy looking, but you look at the root system, this is what a healthy arborvitae root system looks like. It's nice and creamy white. They do have that orange undertone just because they're bark. They have white tips, but along the outside of the ball, these are like shoestrings. They're flat, they're dead. They're kind of a red-purple color. This is Rhizoctonia and Fusarium. So this is actually 2 different fungi. So the way I identify it is I actually grow them out in culture. So this is another arborvitae. Again, canopy doesn't look too bad. Maybe a little sparse here and there, but you know, you'd buy it. You would all buy it. You pull it out of the container. And again, the roots are pretty red. They're flat looking. You get some white tips here and there. Not too bad. But you can even see, yeah, there's a little bit of shadow, but you can see this side looks a little bit better than this. This starts getting a little bit dark. So this is Rhizoctonia and Phytophthora. And so the important thing here is you can't use the same fungicide for true fungi and Phytophthora. It's 2 completely different fungicides. That's why it's really important. So to identify it again, here's the Phytophthora growing here on this plate and the Rhizoctonia growing here on this plate. And this is actually the hyphae, the threads of the Phytophthora that I identified it with. And that's the level you need to go to. That's what has to be done in order to identify these things. You have to have lab work done. So I was kind of curious that I was just kind of looking at the outside of the balls and like, well, what does the inside look like? Because, you know, that may not be representative of the entire root ball, right? So again, the bottom looks pretty good. You know, the orange roots and the white tips. The sides of this ball look terrible. Dug into it, and there's almost no root system in there. But you have to remember, in order to have a disease, you have to have a host, a pathogen, and an environment that favors the pathogen over the host, right? So fungi like cool, moist conditions. So inside that ball is the perfect condition for fungi. So of course they're growing more on the outside surface because the environment favors the host over the pathogen. So this is Rhizoctonia and Fusarium. So this is a dwarf lilac. The root system is not very full. This is like a 1 or 2 year old plant. There's inconsistent growth of the fine roots. So, you know, you don't have them too much. And so this tan color, the white are the healthy roots. And you can see that the tan roots are kind of intermixed within the healthy roots. And so there's definitely a color difference, and the tan roots are a little bit more collapsed, and this is Rhizoctonia root rot. This was the holly that I thought was a really neat example of all these different symptoms, because here's the mass of the root ball, but here's this like one little sprig of healthy roots right over here. You don't see any of that or very little of it anywhere else. So again, when you see strange patterns of coloration and death within a plant, it's usually a disease or it's a biotic factor. This is also Rhizoctonia. This was lavender that I was working with a grower, and he's like, we have a handful of these that are mixed in with our crop. We don't want to have losses. A lot of plant pathology is preventing losses. And so took one of the bad-looking ones, and I was like, yeah, the root ball fell apart in my hand. I'm like, I don't know lavender roots. Like, I don't know what lavender roots look like. So we went and grabbed one of the healthy-looking ones and pulled that out and put it side by side. This root ball stayed together. It had white tips. So it's like, just do this side by side. I'm like, this has an underperformed root system for some reason.
Is there do we know of any correlation between the soil mediums that are used, like vermiculite or pine bark mulch, when these things are grown in that? Do they exacerbate these problems, or does it matter?
It's more of a moisture issue. If you have too much or too little moisture, the roots are going to be stressed and more susceptible to infection. If you have Phytophthora or the oomycetes, so Phytophthora pythium, Phytopythium, they are actually more closely related to algae than they are, and true fungi are more closely related to animals than plants. There's your little factoid. And so if you have the oomycetes, Phytophthora pythium, Phytopythium, if you have excess water, that's exactly where they want to be. They're water molds. And they actually flourish in those situations.
In pine bark mulch, you don't have good water retention.
Right, so that would actually it would reduce your disease incident, and it also acidifies the soil a little bit, and these do not like acidic conditions. So side by side, but the thing is, the one point I want to make with this is that if you cut off this little sprig and let this one go, that looks pretty good, right? It's a healthy looking plant. Is it a disease-free plant? No. So help them, they treated it, turned out to be a beautiful crop. So again, always treat a diagnosis, not a symptom. Because if you treat a symptom, you don't know what's actually you're treating. You can't use the same fungicides, and you're probably not going to get a very good, or you're really just, it's a crapshoot. It's really what it is. Treating a diagnosis, it takes a little bit longer, but again, you can have things either good, fast, or cheap in this world, right? And good, in this case, is accurate. So if you want an accurate and fast, it's not going to be cheap. If you want an accurate and good, or fast, well, you know the combinations. Sorry. I need a slide to do this. But yeah, so if you want a fast and cheap, it's not going to be good. It's not going to be accurate, and that is field diagnosis for all these things. Like, you can definitely say it has a root rot, and if you don't have time, that grower or that homeowner's like, no, we got to treat something tomorrow. Like, they are itching. Then treat them with both fungicides. No problem. Both of them work. You can mix the two. Not a big deal. Prevention is always way worth the cure, because people are getting more sensitive to using pesticides, and that's fine. We need that. We want prescriptive and very knowledgeable use of pesticides. We want to make sure they're used correctly. So don't use unhealthy plants. Pull them out of containers. Look to see if they're healthy. I personally, I buy a lot of plants everywhere, grocery stores, reputable garden centers, less than reputable garden centers. It doesn't matter, I'll buy them, because plants make me happy, I'm a plant person. And so, but I will say that I've been burned one too many times, and I know what's introduced things into my collection, and I quarantine everything, and I kind of napalm them with fungicides, or diagnose them, because I do it when they're in the container. It's so much easier to treat when they're in the container, and then it's not going into our native soils, right? If you know you have a host, or you have a client that has Phytophthora root rot on a plant, and they treat with a fungicide, and you modify the soil for long-term management. So if you drive that soil, it's going to manage the Phytophthora. So you can have a host and a pathogen living in the same place, but the environment has to favor the tree, the host, right? And so the thing is, if you know you have that infection, then you can just not introduce another host into that area, right? So you know you have Phytophthora, look to see what's not a host for Phytophthora, plant that. And then you actually, that's IPM, that's integrated pest management in action, of like, just don't let them come in contact. Landscape management, a lot of it, if you have a root rot, a lot of times drying the soil, I mean, Michelle went on length, I'm not going to talk about soil amendments very much because I think she covered it. But improve that soil environment and sanitize your tools and shoes in between clients or managing stuff. So sanitizing tools, I actually use a toilet brush because it has nice firm bristles, and you can get your shovels and spruce and everything pretty well with that. If you can spray them off with water on site, and then I just use rubbing alcohol, 70% isopropyl alcohol. You can get a bottle at the hardware store, you can get a liter. Go to a hardware store, get a screw-on spray head, so you don't have to worry about labeling the bottle, it's already labeled. And just use that and spray things down to saturation. So if you want to send samples off to have them diagnosed, which I highly recommend, it's a good $15. $15. You want to collect about this is as much roots as you need. You don't need something huge. It doesn't need to be any bigger than a quart sandwich bag or Ziploc bag. You can also plant bleeding cankers, either by using a drill or like a hammer and chisel. And we do, all those tests are initially done by an ELISA test, which I don't have time to go into that. So the University of Illinois Plant Clinic, our state plant clinics, they are the gold standard for diagnosis. If I have questions, if I want to know what's going on in the state, if I want to know what's going on in the United States related to plant health, I go to state lab, diagnostic labs. They are the hub of this wheel. They are the most knowledgeable, they have the best tools to do this. We now have a new diagnostician in Illinois, so it should have a faster turnover rate. But when I was talking to the head of the Texas Diagnostic Lab, a fast diagnosis in plant health is 3 weeks. Because it takes 2 weeks for verticillium to even grow out. You can't make a fungus grow faster. It's 2 weeks, period, no matter what you do to it. And so, so if it's longer than 3 weeks, start getting hit of antsy. If it's less than 3 weeks, that's normal. Don't worry about it. So there is actually a whole network of plant diagnostic labs for all states and territories in the United States. It's the National Plant Diagnostic Network. It actually gets a communication across states, which was originally developed in 2001 because of the concern for bioterrorism. We know something that's killing all the corn in Iowa, we need to know about that really fast, not only because of the economics, but the food and that. So that's why that was created. So in summary, make sure you have things diagnosed. Treat a diagnosis, not a symptom. Help trees during periods of stress, but you have to identify what that stress is. And in the meantime, if you have these problems, plan on treating for two-line chestnut borer. Help that tree get through that stressful period. Help it get on its feet, back on its feet, and let it move on. And it can take care of the secondary problems. Do you have any questions? I know that was really fast. I have 15 minutes to do the next one, so we really don't have time. So I'm not going to take any questions. If you have any questions, please email me or email Tricia and Michelle. We all work really closely together. You can email the plant clinic at the Arboretum, but I need to keep moving. Yeah. Hey, can you do plant diseases in 30 minutes? So the next presentation is on oak decline. Okay. So how many people here have noticed that oaks have been declining at kind of a rapid rate right now? So that is kind of what spurred all this along, because it does seem to be a pretty widespread problem. Talking to people in Minnesota and Missouri and Arkansas, they've already done research on this, trying to understand what's going on down there. I've talked to people in New Jersey, and things are happening over there. And so this is kind of like the aerial view. This is out of Missouri. This is kind of what we're seeing on the large scale, is you have all these widespread death of these trees. And they seem to be happening at a little bit different rates. Some are faster than the others, but we know, with trees, we know that recovery, like if you have construction damage, you won't see above ground evidence of that damage for 5 to 7 years after that damage is done, right? And so you'll have that new driveway put in, and like 5 years later, be like, I don't know why my tree's dying. And you're like, oh, it's the driveway. You have to think in tree time. How long does it take for a tree to recover? So it is happening at different rates. And on the ground, this is what you're seeing. So this is actually, I think, in the Cook County Forest Reserves, where you have a tree that was already removed, you have standing dead that died that year, and you have trees that are starting to staghorn. And it's trying to understand what is all going on to contribute to this. So you have the decline complex when it comes to tree health, And this was actually put together in 1981, and it's still as relevant as it was now as it was then. And so you have these predisposing factors. You have the urban environment is a predisposing factor. There's a reason why Dutch elm disease was so bad, is because these were attracted to stressed trees, right? So genetic potential, soil compaction, poor fertility. You can have, if it's growing out of its range, which we're always kind of pushing things a little bit to their limits. Salt, low soil moisture, poor drainage, climate change, air pollution, and age. Age is seriously a factor when it comes to tree health. I mean, just like human health, we know that babies, infants, toddlers are susceptible to diseases, or they respond to it differently, a lot of times worse. And so do the elderly. It's the exact same thing we see in humans. And so age is definitely a factor. Oops. And so then you have the inciting factors, so you have defoliating insects, you can have mechanical damage like excavation, drought, excessive salt, frost, air pollution, which just, again, keeps putting more pieces of straw on that camel's back. And then finally, you have the contributing factors. So once you get to the contributing factors, these are all the secondary disease problems. So you start getting canker fungi. You get a diagnosis coming back that your trees have like Botryosphaeric canker or Fusarium canker. That is a secondary problem. You haven't identified the primary problem yet. Wood and bark-boring insects, armillaria, verticillium, viruses, and nematodes. And this is just the decline spiral that those trees experience. So what we're seeing here is that these seem to be long-term problems. And so age, species, growth rate, site, and climate. So in the research I've been working on that I'm going to be showing you, we've been predominantly looking at white oaks, bur oaks, and red oaks, just because they seem to be, white oaks especially, are not doing very well. White oaks are very delicate trees. They are very sensitive. While bur oaks are kind of like the junkyard dogs. They can grow anywhere. So also think about your site. If you go to an urban site, I would not recommend planting a white oak. They just don't like it. You have predisposing factors, so slow-growing mature trees, and you have sites that are more prone to environmental conditions. So if you've got an area that floods in the spring, or holds water in the spring just because of drainage in the spring and snowmelt and everything, then you have to take that into consideration. That is a stressor. In Missouri and Arkansas, the predisposing factors that they found were, especially on red oaks, on south-facing slopes, on poor soils, and also with black oaks and scarlet oaks, which isn't really a surprise. If you look at the native environment and the ecology of these species, and especially in the progression of how prairie changes and forest soils change over time, you can get an idea of what these trees actually like to grow in. So you get the inciting factors. So this is a spongy moth larvae, and so you get these environmental factors that start moving in. You get the contributing factors, and so like this is armillaria. These are the rhizomorphs, the shoestrings that you hear a lot about. So this is a really neat example. And armillaria, the neat thing about armillaria is that if there is a stressed tree nearby, trees will get off chemicals, whether they're stressed or not stressed, and so they will communicate that. And these rhizomorphs will actually grow through the soil towards that stressed tree. They are drawn towards those stressed trees. So then you start getting things like hypoxylin canker. So it has two different colors. It has either this nice black color or this kind of whitish bluish gray color. The only difference, same fungus, is that this side is actually sporulating. It's sexually reproducing, so that's why it's a different color. They have changed the name to biscoindoxia, but nobody wants to say that, so just keep calling it hypoxylin canker and you'll be fine. But if you see that name, that's why. Taxonomists need a job too. So the story of oak decline that just working with Gary Watson. So Gary Watson was my boss at the Arboretum for my first 7 years. And so just talking to him, and he's been reflecting over when he's witnessed oak decline since his tenure there. And even back to when he was in grad school. So in the 1970s, they witnessed this, and he was working with George Ware and Virgil Howe, and they noticed oak decline, and their precipitation was 55% above average. And so again, we're going through this period of excess water. In 2017 to 2020, we are 45% above on precipitation. Our precipitation patterns have changed, but we are getting more precipitation. Like we are having wetter springs and wetter falls and these harsh droughts during the summer. So again, in 1980s, They experience above average rainfall, and these were actually the third highest amount of rainfall in 150 years, and they were experiencing oak decline. So this is what it looks like for more recent, so this is 2011 to 2020. This gray line here is average precipitation during this period, or historically. And so here you can see that in all these years you have above average, way above average precipitation. And so again, you think back to what I mentioned with the root rots, or the roots, where roots grow in the upper 16 inches of soil, and it's more shallow in clay soils. And clay soils are poorly drained. And so you get those harsh droughts in the summer, and in the fall and the spring, you have saturated soils because it doesn't drain. It is like the perfect storm. So the observations that we made when we did this study and did the survey to see what's going on, they're mostly in these moderately, poorly, or somewhat poorly drained soils, which I'll let Michelle explain this. So I've never actually had a soil science class, I have to admit. So yeah, but there's this constant variable of poorly drained or moderately drained soils. So this was a site that we visited, the Cook County Forest Preserves, where you have the horribly staghorning trees, kind of the stage 3, 4, 5. There was a tree that was already moved right here. This was 3 days after a rain event, and there's still puddles of water on the soil surface because it's not draining. And so the symptoms of decline, I mentioned the staghorning appearance, even Tricia mentioned the staghorning appearance, and that is your big red flag that you have a serious issue. They start producing epicormic sprouts within the canopy, just like we saw with emerald ash borer, because they are trying to put on growth as much as possible because they want to survive. Trees want to survive. And you start seeing the death in parts of the crown. The leaves may grow in tufts on the branches, and a lot of times when people see stuff like that, they're like, oh, I have oak wilt. And last year, I looked at a lot of trees thinking, that looked like this during the survey. And actually, the only time I isolated the oak wilt fungus last year was from a chestnut. Bad vascular streaking. So the canopy stages, this is kind of what our guideline is for this experiment, just so we can have some sort of reference point. And so if I go to, hey Trisha, are you seeing any stage 2 trees? And she'll be like, I'm out all the time. Yeah, I'll keep an eye out. And it's like, okay, I'm looking for sites that have trees like that. But it's just the slow progression. So you get those 1/2-inch twigs that are starting to be uniformly exposed. Then you start getting the 1 to 2.5-inch branches, and then those largely die out. And by the time here, a lot of those branches on some species are brittle and they've already broken off. But all the growth is down here on the stem. So the 2021-2022 Oak Decline Survey, it was folks at the Arboretum, city foresters, forest preserves, park districts, arborists. We went on to private property. Some homeowners let us include it on their survey. A lot of street trees, cemeteries, park districts, forest preserves, all across the board. Again, looking at those 3 species. And we tested all these root trees for these 3 species, Phytophthora and Phytopithium. So these are the oomycetes. They are not true fungi. I'm not going to call them fungi because that just confuses things. They are oomycetes. And they are favored by wet conditions because they are water molds. And so here's a brief map of where we surveyed, at least in 2021. No, this was 2022. And the stars are where we found declining trees and they tested positive for Phytophthora. So again, it's all across the Chicago region. But the thing is, you have to remember is that there are some native Phytophthora. They happen. We know they're there. So one of the big questions is what we're dealing with a native problem or is it an introduced problem? So even at the Arboretum, we sampled, and even out in our forested areas where there's not many introductions occurring, and still got positive trees. And we also kind of look at the topography, because again, you have to look at drainage. And so we had mostly flat topography with little water movement, slow percolation through the soil, because we have clay soils, and seasonal water tables are usually pretty shallow in these conditions, in these sites. Oh no! All right, yes. So from the results of 2021 survey, so this is actually what Phytophthora looks like, or at least one species. It is kind of pretty, it's like a snowflake. So we surveyed 87 trees from these different sites and tested them. They came 78 trees, 78 declining trees that had those canopy symptoms, 83% tested positive. And 42% of healthy appearing trees also tested positive, which is, again, very interesting. So this goes back to the disease triangle. Most of these trees that had symptoms were kind of in bottomland areas. They were in areas that were poorly drained. A lot of these trees were kind of in upland species that were a little bit, or sites that were a little bit drier. You have to have all three variables to have disease. You can have infection without disease if the environment favors the host over the pathogen. So again, the big question is, so Phytophthora seems to be really prevalent, and we kind of notice those variables that kind of lead into those symptoms of poorly drained soils. You have clay soils, you have flat topography, you have older trees, which are those are the trees that we really want to save because those are really, they're cool and valuable ones. That's going to take a long time to get more. And so the next question is, what species are we actually dealing with? Are they native species? Are they invasive species? So I'm working with Frederick Miller. He and I got a DNR grant where we're going to be surveying all the public lands across Illinois to see, because he's seeing it across the state, because he does all the, he does some of the gypsy moth or the spongy moth trapping, but walnut twig beetle and all those. And so we're going to be taking roots from those, healthy trees and symptomatic trees, and seeing what we're seeing across the state, and comparing to what we're seeing. And so we actually are growing out the Phytophthora, or the oomycete. So this is what it looks like. We have to put it in a shaker, so they kind of look like little Dippin' Dots. Yeah, you're going to look at those all different from here on out. And so we're actually doing the DNA extraction and sequencing. We're setting up the Oklahoma State University, and we're going to see which species is actually growing. So hopefully in this next 6 months to a year, I'll have those results and we'll have a better understanding what's going on. So in the short-term management of everything, especially if you're dealing with landscapes, when you're dealing with forested areas, it's a tougher situation. So you can go in and manage all the soil and treat a bunch of fungicides. It's just not possible. Forest management is a different system than landscape management. So if you're dealing with the landscape management, you're going to put out the immediate fire. So you're going to have the tree diagnosed. If it has a Phytophthora root rot, you're going to treat that fungicide, right? That puts out that immediate fire. It'll calm down that system and hopefully prevent all those secondary problems from moving in. So again, send samples to the University of Illinois Plant Diagnostic Lab. If I get too busy in my lab, I send it to them. We collaborate quite a bit. So they are the resource to go to. You send in samples, like I mentioned before. And so then in the landscape, on established trees, you can treat them with fungicides. And there are fungicides that are available for Phytophthora. Usually they're either a phosphate base, so like Phosphatyl AL, Reliant, any of those, or they're Subdue Max, which is mefenoxam. So do that. If you bring in new plants, quarantine them, and if they are infected, treat them while they're still in the container. Plant healthy plants. Healthy plants, healthy landscapes, healthy planet, right? And nurseries, if you work with nurseries, always check nursery stock. Pull it out of the container, cut into the root ball if you can. Nurseries sometimes have policies that you can't disturb the root ball. Again, would you buy a house and be like, no, just don't go in the basement, we want to buy the house, right? You're like, I don't think I do anymore. You're buying the entire thing. You wouldn't buy a car and not drive it, right? You're dealing with a living organism, and you want it to live for a really long time. You want to be in good condition. Like, we always make assumptions that nursery stock are always healthy. And this is not to knock nurseries, because I come from a long line of farmers. I really care about those primary producers. This is their livelihood. It feeds their kids. But they also need to be producing healthy stock. And their own standards, the ANSI Z60.1 standards, say that those plants should be healthy and disease-free. So, again, look at them. And then also give those nurseries feedback. Hey, I bought this tree, got it diagnosed, had this. And because if they don't get that feedback, they don't know what problems they have. You have to have that feedback loop so they can start cleaning up their act. And everybody will be better in the long run if we help, educate nurseries too. Long-term management plans. So once you put out that immediate fire, you put down fungicides, and managing these root rots in our native soil can be tough. Because if you don't modify the soil, that environment will still favor the pathogen over the host. So you have to also implement long-term management. And so this is where soil management comes in. So air spading, adding in compost, changing drainage, moving where that downspout is to somewhere else. Anything you can do to keep water away from that plant if it has a Phytophthora or an oomycete root rot. Don't blindly water. That's what had one of our instincts in the green industry is when in doubt, water and fertilize, right? Like, I don't know what's going on, just water and fertilize it. We need to get away from that. Don't blindly water. One of my favorite tools to use out in the field is a hori-hori, or a soil knife. Everything I collected over here, I mean, it really is, it's like a steak knife. It will cut you. And I know from experience. But no, this is one of the best tools. But people, when I talk about, hey, well, how do the roots look? You have a declining tree. They're like, oh, I didn't have an air spade. I'm like, you don't need an air spade for this. You need a tablespoon to go out there and dig up. Like, use your hands. You can dig up. You can dig 10 inches down in the soil very easily. But don't blindly water. Either dig, see how moist the soil actually is, at least 10 inches down. You get a soil probe. Which I don't know if Michelle has one, but spend $45 on a soil probe. It will help you a lot. And make sure you're watering for the plant species and the soil type, because drainage affects a lot of this. So treatments, so yeah, you always have to identify the primary stressor, reduce exposure to those factors, and then start getting that plant to restore. So I'm actually 1 minute over time. I'm really sorry I left it over. Yeah, but I like to go on. And I'll stay for maybe 10 minutes afterwards. I know probably some of you need to get going. I'll stay for about 10 minutes to answer questions. Otherwise, thank you so much. This has been a wonderful event. The organizers want to do you have something to say?
Just really quick, if anybody would like their CEUs, you need to sign the sheet and take one of these. So see myself or see Lori before you leave.
All right, wonderful. Thank you.
Thank you.
Have a nice day. Yes.
Yep.